Hey guys, welcome back to the
Swaggy Science
Students
Channel!
Channel!
And today we're talking about cellular respiration and classical genetics.
So our first topic is meiosis. So the first thing we'll be talking about is chromosome numbers. So our first vocab word are genes. So genes controls inherited trait expression. So that's like what color eyes you get, if you can produce insulin or not. And then from that, we'll be talking about homologous chromosomes. So the number in humans is there are 46 individual chromosomes, and so there are 23 pairs, and each parent combines their 23 chromosomes to offspring. So there's 23 from the mother and 23 from the father.
Combined to be 46.
So homologous chromosomes are paired chromosomes, each having identical genes.
Same.
Okay, now we'll be talking about gametes. So gametes are sex cells, which is like sperm and eggs. And these cells are first haploids, and haploids are having the number of chromosomes. So when the sperm and the egg meet, it's 23 and 23, so that equals 46 total chromosomes. And then this
So haploid is half of them.
Yes, and this is like happens through fertilization when the gametes meet. Yes, like that. And diploids are having the full set of chromosomes.
46. Okay, so I'm going to talk about meiosis the first time through. So in this set, you go through meiosis twice, there's different stages. So meiosis is the formation of gametes, which he just talked about. And it reduces the number of chromosomes in the sperm and eggs. So the result is different copies of haploid cells. So in meiosis, you're trying to make different cells, whereas in mitosis, you're trying to make two identical cells. So the stages are very, very similar to mitosis. So the first stage is interphase, which is exactly the same as growth and metabolism. And then in prophase 1 is the pairing of the homologous chromosomes, which is basically they're crossing over. The vocab word is crossing over, which is segments of chromosomes exchanged between chromosomes during meiosis. And then in metaphase 1, homologous chromosomes line up at the center, so the midline, like the equator. And then anaphase, the same chromosomes separate and move towards the poles, which is the same. And then in telophase, the spindle fibers break down and the chromosomes uncoil into their relaxed form of chromatids, and then they form the two nuclei.
And that's the thesis.
You would, Aaron. So now I'll be talking about the second division of meiosis. So the stages are prophase 2, which is when the chromosomes condense, and then the spindle reforms and attaches to the chromosomes. Then metaphase 2, the chromosomes line up at the equator of the cell, which are single X chromosomes. And in anaphase 2, sister chromatids separate and travel to poles. Then in telophase 2, the spindle breaks down and 4 nuclei form around chromosomes and the cell divides. The results of meiosis 1 and 2 are 4 new cells have formed from 2 divisions, and each nucleus contains a haploid number of chromosomes, so 23.
Yeah, so like in the first round they create 2, and then in the second round they create 4.
Now we're going to be talking about the importance. Meiosis is very important, so we don't all look the same and we are all unique in our different ways. Crossing over also helps with the variation of DNA. Next, we'll be talking about sexual versus asexual reproduction. Asexual means that all the chromosomes come from the parent.
One single one.
It is the exact copy of the parent. There is no genetic variation. Sexual reproduction is each parent contributes an assortment of chromosomes, which is crossing over. So 23 from Ma, 23 from Pa, checkity check.
Bing,
Genetic
bam, boom.
variation equals a healthy species.
Hey guys, welcome back to part 2. Today we're talking about Mendelian genetics.
So the first part to Mendelian genetics is the beginning. In 1866, Gregor Mendel, who is the father of modern genetics, he was an Austrian monk and had a good math and science background.
And what he did is he used pea plants to figure out like the different traits
So he found out which ones were like the fastest growing, which were traits that he could easily see, and he used to manipulate fertilization and observe the results from that.
So like if the plant was yellow or green, the pea.
Or tall or short. The second part is the inheritance of traits. Starting with purebred plants, a capital P generation would be a parental or a self-fertilization. He then cross-fertilized the parental genetic plants, which would be like F1 and F2 generation. Results of the lower P generation cross would be F1 generation, which equals all peas were the yellow color and green peas disappeared.
Yeah, they weren't there.
Mendel then crossed plants of the F1 generation, and then green peas reappeared. The ratio of yellow to green would be 3-1.
Okay,
So it looked like this. This is the pea generation, then this is the second generation after that, and the third one when the green ones came back.
Okay, so genes in pairs. So Mendel, he realized that something must control traits, so we see the factors. So each pea
genes.
plant must have 2 copies of a trait, so one from each parent. And the factor genes come in pairs. So an allele is a form of a trait. Dominant alleles are the dominant in it and are represented with a capital letter. So let's say there was yellow and red. Yellow was the dominant, red was the recessive. It'd be a capital Y for dominant.
Yep.
And then alleles pairs must have the same letter. So like in that example when I did yellow and red, the red would be an undercase Y because it has to match the letter.
Otherwise, it would be a different trait,
Yes.
not the same allele.
Okay, so the next part is dominant. So the first generation peas are same yellow as the parental peas. So different alleles make up the F1 peas. So the parents could have it's meaning that all of them were dominant, so they all came out as the same color. But they could have different genes from the mother or the father could have different genes that show up later. So homozygous they both have the same letter, so it'd be capital TT or undercase TT, which is saying that it's yellow dominant but dominant all the way through, or yellow
Pure red.
Yeah, pure red. Or it's the recessive gene all the way through. And then heterozygous different, so it could be one dominant trait and one recessive trait.
And the dominant trait is placed first.
Yes. And then the next part is genotypes and phenotypes. So the genotype is the actual makeup, so the actual pairs. So uppercase Y and lowercase y are the genotypes, so that's what they are. And then the phenotype is observed characteristics, so what they look like. So that would be brown hair, blonde hair, and all of that.
Okay, so I'm going to talk about segregation. So the law of segregation is 2 alleles for each trait separating during meiosis. So the uppercase Y and lowercase y. The uppercase Y would come from one parent, and the lowercase y would come from another. So, but then after the fertilization, the offspring would have an uppercase Y and a lowercase y. So the gametes give 1 allele each. So the father would give the uppercase Y, and the sperm would give the uppercase Y, and the egg would give the lowercase y. And then when they're fertilized, the offspring would have uppercase Y and lowercase y. And that's called a hybrid or a heterozygous organism. So I'm going to talk about what a monobrid cross is. So that's when you cross one pair of contrasting traits. So to do this, you would use a Punnett square, which helps predict possibilities of the traits the offspring will receive. So in a homozygous, things are the same. So if you cross 2 homozygous alleles or offsprings, for example, if you crossed a yellow pea pod with with a green pea pod, but they're both heterozygous or homozygous, that means that the yellow genotype will be uppercase Y, and the green homozygous genotype would be lowercase y, lowercase y. So the phenotype of both of them would be yellow and green. So the first step you do is you take your square, which has 4 squares in it, and put one parent on the left side and one parent on the top side, and then you fill the boxes with the combining alleles. So for an example, in the homozygous, it would be like this. So the mother, the uppercase Y and the uppercase Y, and then the lowercase y and the lowercase y. So you can see in the thing, when they cross, they're no longer homozygous alleles, they're heterozygous because they have one uppercase Y and one lowercase y. So the results from this would be the genotype is uppercase Y, lowercase y. But you would have 4 yellow plants because yellow is the dominant gene, and there's uppercase Y in all of the genotypes. Let's not do this.
So now I'll be talking about independent assortment. The law of independent assortment is the random distribution of alleles. The inheritance of one gene does not affect the inheritance of another.
Okay, so I'm going to talk about dihybrid crosses. So we're going to cross a homozygous round and yellow with a homozygous wrinkled and green. So the round and yellow is dominant, and the wrinkled and green is recessive. So in the dominant one, you have uppercase R, uppercase Y, uppercase Y, and then lowercase r, lowercase y, lowercase y. So that means what we're going to do is called FOIL. So FOIL is the first, the last, the outsides, and the insides. So you take your genetic genotype and you do the first one, which would be A and B, the last ones, the middle ones, and the outside ones. Once you do that, you place all 4 genotypes you have. So like the genotypes would be uppercase R, uppercase Y, uppercase R, uppercase Y, etc., or lowercase y, lowercase r. So you would then place it on a Punnett square, but instead of it being 4 squares, it's 16 squares because you have 4 genotypes from each parent. So the setup looks like this, and you take the R's and they go first, and the Y's and they go first, and then you just do it with like you do a normal Punnett square and set it up all the way, with one parent on the top and one parent on the left, and each will have one allele for each trait. So with this example that I just showed, the result is 16 genotypes with uppercase R, uppercase Y, lowercase y. So the phenotype would be 16 yellow round pea plants.
Now, we'll be talking about probability. So what probability is the likelihood a specific event will occur. So it's the number of all the events. We're going to be talking about recombination. So genetic recombination are genes produced by crossing over and independent assortments. So it's 2 to the n, so it's the number of chromosome pairs. So the genetic recombination of humans is 2 to the power of 23 times 2 to the power of 23, which comes out to be like 70 trillion genes is the makeup. So that's the male, which is 2 to 23, times the female, which is 2 to 23.
So in that, if you had 70 trillion children, none of them would look the same unless they were twins, and then the 70 trillion and one would be an identical copy of the first.
So there's no one ever in the world that's going to look the same because there's not 71 trillion people in the world.
Now I'll be talking about linkage. So some genes that stay close and wind up together. So chromosome maps are gene sequence on chromosomes that use crossover data. Map percentages equal sequence position on the gene.
Next, I'll be talking about polyploidy. That is when there are extra sets of chromosomes in an organism. It is very common in plants and can be lethal in animals.
An example that would happen in plants would be crops like corn, and it doesn't really make a difference in plants, but like in animals, like she said, it can kill them.
So that's all we have on this part of the Science
Chapter
Students.
10. Bye guys.